Consider this case file. A 27-year-old female patient arrives at a gynecology-oncology clinic with a single, alarming primary complaint: severe, persistent vaginal bleeding that has lasted for an entire year. Upon physical examination, the source of the hemorrhage is immediately apparent. There is a massive 5 to 6 centimeter tumor deeply involving the upper vagina and the surrounding cervical tissues. Our objective is to trace this exact macroscopic clinical presentation backward. We are going to reverse engineer this tissue failure down to its microscopic cellular origin. In oncological clinical thinking, a patient's physical symptoms are a direct, readable map pointing to a specific underlying cellular dysfunction. This severe bleeding represents the mechanical endpoint of a highly organized biological invasion, one that initiated years prior in a very specific anatomical vulnerability. To understand that vulnerability, we have to look at the normal anatomy of the cervix. Specifically, we need to focus on a precarious boundary region where two entirely different tissue environments meet. This diagram illustrates the squamo-columnar junction. It is the precise geographical line where the flat, multi-layered squamous cells of the protective ectocervix meet a single layer of tall, mucus-producing columnar cells from the inner endocervical canal. This specific border region exists in a constant, active state of cellular remodeling and regeneration, known as squamous metaplasia. Squamous metaplasia is the physiological replacement of one fully differentiated cell type, like columnar cells, with another type, like squamous cells, to handle environmental stress. This constant architectural turnover requires the foundational basal cells to divide rapidly. Because they are dividing so frequently, their genetic material is repeatedly exposed to the outside environment. Consequently, the cervix's own necessary ongoing architectural remodeling creates an area called the transformation zone. This zone serves as a vulnerable back door for an oncogenic virus to hijack. The primary antagonist here is persistent infection by high-risk human papillomavirus, specifically the aggressive oncogenic strains HPV-16 and HPV-18. The precise mechanism of entry is insidious. The virus integrates its own viral DNA directly into the host cell's native genome. To understand the damage, consider the host cell's normal internal guards, the critical tumor suppressor proteins p53 and Rb. These are regulatory molecules that normally monitor DNA health and stop chaotic cell division. This animation shows the viral counterattack along the host cell's DNA strand. The integrated HPV DNA begins expressing two highly specialized viral proteins known as E6 and E7, which target, bind to, and dismantle those p53 and Rb guard proteins. The loss of p53 and Rb is devastating. The cell loses its ability to initiate apoptosis, programmed cell death, or to halt the replication of mutated DNA. By neutralizing these genetic guards, the virus renders the cell blind to fatal mutations, forcing it into an unstoppable cycle of chaotic, unchecked replication. The first consequence of this viral sabotage is epithelial dysplasia. Dysplasia is an abnormal development of cells where the previously organized tissue layers begin to lose their uniform structure and orientation. This diagram illustrates the progression into cervical intraepithelial neoplasia, or CIN. Here, the mutated cells begin multiplying at the bottom layer and stacking upward, displacing the healthy cells above them, while the bottom boundary line remains completely intact. Up to this point, a critical physical barrier has kept the danger strictly localized: the basement membrane. This thin layer of matrix separates the epithelial lining from deeper connective tissues. But then we reach the tipping point. The chaotic cell mass secretes enzymes that break through the basement membrane, spilling aggressively into the deeper stromal tissue. This breakthrough defines the exact moment the condition graduates from a manageable local neoplasia to a lethal invasive squamous cell carcinoma. We can now map this microscopic invasion directly back to our patient's primary symptom, the year-long severe vaginal bleeding. The mechanics of the hemorrhage are physical. As the invasive tumor tears into the deep cervical stroma, it crushes and destroys the normal local capillary architecture. To sustain itself, the tumor forces the rapid construction of its own defective blood vessels. These new vessels are incredibly fragile, leading to spontaneous bleeding upon slight mechanical trauma. The secondary symptom of abnormal fetid discharge connects directly to the tumor's growth rate. The mass outpaces its own blood supply, causing the center to die off in a process called necrosis, which triggers intense local inflammation. The origin of advanced pelvic pain is similarly mechanical. The invasive mass pushes outward beyond the cervix, into the perimetrium, directly compressing major pelvic nerve plexuses. When a doctor conducts a physical speculum exam, these internal mechanisms correlate with what they see: a friable, ulcerated mass that is exophytic, meaning it grows outward beyond the surface epithelium. The bleeding, discharge, pain, and ulceration are the direct mechanical physical manifestations of a microscopic cellular invasion destroying tissue architecture. Oncologists utilize a specific diagnostic matrix of paraclinical testing to prove the invisible viral sabotage we just tracked. The Pap smear acts as a cellular dragnet, sweeping the surface to capture the dysplastic cells and abnormal nuclei shed from the exterior of the growing mass. The HPV DNA test bypasses the physical symptoms entirely to look for the exact integrated viral genetic material hiding inside the host cells. This slide shows definitive proof: the histological biopsy. It visually confirms the invasion of dense pink clusters of malignant squamous cells deep into the cervical stroma. Looking closely at structural markers like keratinization identifies the tumor strictly as squamous. These lab results represent the physical, genetic, and visual receipts documenting the tumor's exact origin and mechanism of destruction. Left unchecked, the forward trajectory of the disease follows a brutal logic. It progresses from localized cervical invasion outward into the perimetrium, moving steadily into the pelvic lymph nodes, and finally achieving distant systemic metastasis. Envision the cervix as a critical structural gate. Originally, this gate is protected by dedicated genetic watchmen, the proteins P53 and RB. The high-risk HPV virus sneaks through this gate and deploys its saboteurs, E6 and E7, to eliminate those watchmen. This leaves the gate entirely defenseless against a chaotic cellular rebellion. Mastering the pathology of cervical squamous cell carcinoma requires looking past the macroscopic symptoms of bleeding and pain to recognize the unbroken, continuous chain of causation stemming from a single viral integration.